The overall goal of the following experiment is to measure the effect of environmental compounds, such as different types of bacteria, on C. elegans egg retention. This is achieved by first carefully selecting L4 stage larvae to ensure worms used in the assay are exposed to the desired environmental cue at the beginning of the adult reproductive stage. As a second step, L4 larvae are placed on lawns of different bacteria for a fixed period of time in order to expose worms to the cue during their peak egg production and laying period. Next, worms are bleached to dissolve the outer cuticle and expose the eggs. The number of eggs that were retained within the uterus are then counted. Results are obtained that show the number of eggs retained varies based on the different types of bacteria fed to the worms.
Though this method can be used to determine the effect of pathogenic bacteria on the C. elegans egg-laying system, it can also be used to determine the effects of other compounds, like environmental toxins, on the egg-laying system of this common model organism. Demonstrating this procedure will be Mary Rosell and Mona Gardner, both students in my laboratory.
To prepare agar for 50 plates, first add 1.5 grams of sodium chloride, 8.5 grams of ultra-pure agar, and 1.25 grams of peptone to 487.5 milliliters of deionized water in a 1-liter flask or bottle. Gently swirl the contents to combine them. Sterilize the solution by autoclaving. Once autoclaved, remove the flask and allow the solution to cool for approximately 1 hour in a 45 degrees Celsius water bath. Next, add in the following order to the cooled agar solution: 500 microliters